So next we have Dr. Chitra Ravi. She's a postdoctoral research fellow in our group at UQCCR, and she is going to talk to us a bit about metagenomic-based diagnostics for bloodstream infections. But she's recently completed her PhD looking at neuropathogenic E. coli.
Thank you for the invite and for the opportunity to present some of our work today. So I'm going to be talking about accelerating pathogen and AMR detection directly from blood using metagenomic approaches. So bloodstream infections, they are basically the presence of viable microbes in the blood that can lead to sepsis, which is a life-threatening organ dysfunction caused by a dysregulated immune response. So sepsis has a very high mortality and morbidity rate. So approximately 25 million sepsis episodes occur in children annually, leading to around 3 million deaths. So the time to getting treatment is extremely crucial here, but the reference current diagnostic methods are culture dependent, and they do have a few limitations, and that includes that only around 30% of these blood cultures are positive during these cases, and prior antibiotic treatments can interfere with the detection of these pathogens. And lastly, there could be a long turnaround time for blood cultures to be flagged as positive and to also get some results from antibiotic sensitivity tests. So the entire process of growing it in culture and then trying to get a single colony and then doing gram staining and species identification, followed by antibiotic susceptibility tests, can take up from 2 to 5 days, especially if they're slow growing. And during that time, the patient mortality increases significantly. So if we can reduce that entire period to maybe 24 hours, if I'm being a bit too optimistic, that can really improve, early treatment and basically improve patient outcomes, basically. So at the same time, we have actually started to do a clinical trial. So it's called the DECA study. And our aim for this study is to inform future design and implementation of rapid bloodstream infection diagnostic methods. And our study population would include children from as young as 1 month to 18 years under oncology care with suspected sepsis. So the collection site for these is from the Queensland Children's Hospital from both the oncology and ED wards. So we collect blood samples immediately when we suspect they have sepsis, and we also get blood culture. So we get blood culture through standard of care. We also get in some whole blood. So with the whole blood, we are doing some DNA extractions, and we also spin down the whole blood to get plasma to get cell-free DNA. So cell-free DNA is basically just shorter fragments of DNA that's circulating outside of the cells and have shown that they are a good sample type for diagnostics. And we're also collecting some serum for any future antibody detection work, and as well as PAXgene tubes for future host transcriptomic studies. So we've got all these samples, and we're collecting a lot from these children. So there's a few things that we want to take note and keep in mind when we're doing all this work. So firstly, the quality of the data that we're going to get, and it has to be agnostic, broad detection of all these pathogens, user-friendly, so that anyone could eventually adapt and be able to do it. It shouldn't take too long, a reasonable cost per sample, accurate, sensitive, easily adaptable in any lab, and hopefully ease of clinical validation and integration into pathology labs. So that comes down to which approach we could use for this. So that's clinical metagenomics, which has shown to be quite promising. So what this means is that you get a clinical sample, be it from whichever site from the human body, and there's a few steps. So the first step is human DNA depletion, which you could do. What this does is just depletes for human DNA, because most of these samples have over 99% is going to be human DNA, and that could affect the usefulness of your sequencing data. So after that, you can go ahead and extract your microbial DNA and then go on to do your library preparation and sequencing. So that sounds easy when it's all put down in bits like this. But there is quite a few challenges when using blood sample as our matrix. So blood has a lot of things inside it. And a lot of these things can obstruct sequencing PCRs. So for example, hemoglobin releases iron, there's lactoferrin, IgG, and heparin that can inhibit PCR. And the other crucial thing for us is that the bacterial load during bloodstream infections are very low. You get around 1 to 10 CFU per mil. And if you want to look at genome copies from both live and dead bacteria, that's around 10 to the 3, 10 to the 4 genome copies per mil. And that's a huge difference when you compare it to blood culture because you're enriching for it. So those are the challenges that we have when using blood. So there's a few workflows that we're trying out to overcome these challenges. Firstly is usually using a shotgun sequencing method from both of the two very popular sequencing platforms, Oxford Nanopore Technology, ONT, and Illumina. So ONT does long read sequencing, Illumina does short read sequencing. So we basically get some blood from volunteers in our lab and spike it with a known bacteria in known concentrations, and then go on to test out a few of these extraction kits that has both human DNA depletion and microbial DNA isolation, or kits that does total DNA extraction. And then as a little quality check for us to see whether we're able to identify the pathogen we spiked it in with, and also to assess for any human depletion, we just do a qPCR, and then eventually move on to library prep and sequencing. So I have tested a few of these kits and have done qPCR, so I'm just going to show you some of the results for that. So I started out with E. coli and spiked it into whole blood from a high bacterial load of 10 to the 8 down to 10 to the 1. And I've also included a negative unspiked blood control. And because our cohort is children, it is highly likely that the blood samples we're getting is going to be less than 1 ml. And so far we have been getting less than 1 ml, or maybe 1.5 ml max. So we need to make sure that whatever method we have is going to be sensitive enough for low blood volumes. So we have tried that out with different volumes of blood. And in the table here are some of the kits listed that we have and are currently testing. So in the second column here, so these first 3 kits have both the human DNA depletion steps, and then it goes on to DNA extraction. So it's a differential lysis. And then the last one has will extract both the human and microbial DNA. So you could do some modifications afterwards to get rid of the methylated human DNA, or maybe just sequence everything all together. So for cost per sample, the first kit is the cheapest, $7 per sample, and the Molzyme molasses kit is the most expensive, coming to $75, mostly because it is from Germany and it needs to be shipped with dry ice, so internationally and domestically, and that piles up. So in terms of extra steps, we've got Zymo kit here that has a blood lysis step, so you need to buy that reagent external to the kit. In terms of processing times, the molasses kit was the quickest. So this is time taken for 10 samples. If it's just 1 sample, it's just 2 hours. So it's very quick. The last 2, I've yet to try that out. In terms of ease of processing, the molasses kit was definitely the easiest to follow, and for anyone that's new to extractions, I feel they can learn it quickly because the manual is very detailed and you'll know each step and the purpose of each step. So that's something I appreciated. So I had gone ahead and done qPCR. With MP Bio, I actually had to pause that because it was not, it was pretty hard to process the samples and it got stuck in the filters. So I had to take a pause for that one, and I continued doing qPCR for these 2 that are highlighted in the red box. So on the left here, we've got the qPCR graph detecting for E. coli. So it's plotted against the CT value. So the higher the CT value, the lower the amount of starting DNA that you've got in your sample. And it's on the x-axis, we've got the sample type. So on the left here with the black dots are just the controls. We've got the E. coli genomic DNA, the human DNA, and water. And then we've got the dilutions here from 10 to the 8, 5, 3, 2, 1, and the negative unspiked blood. So the molasses kit is in blue and the Zymo kit is in red. So obviously we see a trend of the lower the bacterial load, the higher the CT value, which is good. We did notice that at the point of 10 to the 2 and 1 CFU per mil dilution, we were only able to detect for the E. coli from the molysis extraction kit and not from the Zymo kit. So molysis was far more sensitive compared to the Zymo kit. And then on the right here is for the human DNA detection, because these kits did have the human DNA depletion step. So in terms of control, we only detected for human. That's good. And across, we did see that the Zymo had a larger CT value. So it means that it depleted more human DNA. So there's lesser in the sample. So it did have a better depletion compared to molysis. But at this point, we really wanted something that was very sensitive because during bloodstream infections, you get low microbial load. And we also have low blood volumes to work with. So we stuck with molysis for now. And I've also tried out different volumes of spiked blood. So we've got 1 ml in dark blue, followed by 0.5 ml and 0.2 ml in the lightest blue. So we've got the dilutions down here, 10 to the 8, 5, 3, 2, 1. And in 10 to the 1, we could still detect for 0.5 and 1 ml, but not the 0.2 ml. So it seems that 0.5 ml is the minimum required for at least 10 to the 1 CFU per ml. So that looks promising. But that being said, this is a PCR. Sequencing it, we might get a different data set, so that's something we need to look into. So the next approach that we wanted to try out is from this company called Micron Brain. So this company has a Devon host depletion filter, which is shown here. So basically, the entire idea is as you're getting blood from the hospital bedside, you pass the blood through that filter, and what it does is it depletes for human cells, like over 90% of human cells. And what passes through is basically your microbial community unaltered. So that's an immediate human DNA depletion technique over that, which is really nice. And then you go on to extract your microbial DNA, and then they also have a library preparation kit that has been optimized for very low input samples. Because they're aware during bloodstream infections you have low bacterial load. And then you go on to do sequencing. So it's pretty quick. According to their pipeline, so they also have a bioinformatic pipeline called the PartySeq. So if you do all of this and do sequencing and with the pipeline, they have said that you can get results in 24 hours from blood collection. So that's really quick. And the last one is the hybrid capture sequencing. So that involves using panels. So panels are basically probes that have sequences of the pathogens and of the AMR markers. So it latches onto those DNA. It's like an antibody latching onto your pathogen. So we've got both panels, one from Illumina. It's a commercial panel called the RPIP. So it's the Respiratory Pathogen ID/AMR Enrichment Panel. So it detects for over 180 bacteria, I think 50 viruses and 40 fungal, and over 2,000 AMR markers. And then we also have Twist panel, which was custom made. So Twist provides that service of custom making panels. So we have a more sepsis-focused pathogen bacterial panel from Twist. I think it detects over 60 bacterial species and over 3,000 AMR markers. So for these, we were trying out two different sample types: spiked plasma. So basically getting plasma and spiking it with known concentrations of shared DNA to mimic cell-free DNA. And then we also got spiked blood. And then we go on to do extractions to get the DNA, and then library preparation, which involves the hybridization step. So this one is a bit longer library preparation-wise as compared to shotgun sequencing, but has shown to be more sensitive in literature. So that's something we're keen to compare and try out. I have done some work with the RPIP panel. So the samples I've used is spiked plasma. So we had a few clinical samples in the freezer, and we wanted to test it out. So we used 3 ml of clinical plasma sample that has shown to be negative for blood culture, and we used that for cell-free DNA extraction. We did want to use our own plasma, but the thing with being healthy is that you don't get enough fragmented cell-free DNA if you're healthy. So we had to try our luck with a patient sample. We then went ahead with spiking it with 10 to the 5 and 10 to the 6 genome copies of shared E. coli DNA. So usually during bloodstream infections, you get 10 to the 3 and 4, but we wanted to push it up just to see if this method works. And then our next sample type that we wanted to try out was with the frozen clinical blood samples. So it was obtained from another study called the Duration Study, and it's adult blood samples, and it had already been used for another project, so we had varied volumes left over in the freezer, and I thought just give it a go and see what happens. So analysis-wise, I'm not very bioinformatic apt, so I just went on to Illumina BaseSpace that has 2 applications, which is xAmplify that creates a clinical report, and the Dragon Microbial Enrichment Plus, which gives you a more detailed report in terms of the sequencing depth and the reads. So I'll just show you some of the data from that. So on the left here, we've got, so this one's for the spiked plasma samples. So on the left, we've got composition of reads from both the 10 to the 5 sample type and the 10 to the 6 in these pie charts. So for both, we were able to identify the pathogen that we spiked with, which is E. coli, with a few contaminants in there. So, and the other thing is that obviously with the amount of DNA, the more DNA you put in, and the more DNA you put in, proportionally you get higher amount of reads. So that looked good for us. And then on the right, we've got these bar graphs with all of the AMR markers that we that are associated with E. coli. So in the pink is the number of reads of these AMR markers from the 10 to the 5, and in the blue it's 10 to the 6. So we did identify all of the associated AMR markers in E. coli, and the most important one, which is the extended spectrum beta-lactamase CTX-M15, so which was great. We also saw the same proportion. The more DNA you put in, the more reads you get. So this one is from the frozen clinical blood samples. So we use the RPIP with this as well, and we were able to detect 5 out of 6 of these samples accurately. So the column here is the pathogen identified previously using microbiological technique like blood culture, and also using T2 Biosystems, which is a PCR magnetic resonance technology. So they both identified the following pathogens here, and this column is what we had detected post-sequencing. So it managed to identify all of it except for sample 6 accurately. And I'm just going to talk about the first 5. We did identify, it did have a percentage identity of over 99% for the first 5. With quite a fair few aligned reads. It did vary, and I think it varied probably because of the amount of volume I had left of these blood samples. It were not the same. Some were as low as 600 microliters, some were as high as 4 mils. So that could be the reason why. And also we did identify the AMR markers, but there's one key one that we were missing that I'll get to. So for sample 6, we did identify Salmonella enterica instead of Staph aureus with a percentage identity of 98.6 and a significantly lower line reads, but still substantial. We didn't identify any, MSSA with a lower number of reads at all. It was just this one. And we did see that the AMR markers identified corresponds to gram-next rather than the gram-positive, which is what Staph aureus is. So there is a discrepancy between the molecular microbiology analysis with the sequencing data. We did go back and check the blood cultures and anything clinical-wise we had checked, and we made sure nothing was mixed up. So for now, we can't really describe what had happened and what had caused this discrepancy. And the other thing we realized is that we were unable to detect for the MEC-A, which provides the methicillin resistance in Staph aureus. That could be 2 things. One being these samples were not of the best quality. They have been freeze-thawed a couple of times, different volumes. And the other thing is we might need to amp up our sequencing depth, like the amount of reads. So to get information such as AMR, we need to actually increase our sequencing depth. So that's a lot of work and money that goes into if you want those finer details. So the work still looks promising, but if we do want to hopefully eventually get it clinically implemented, so there are some limitations based on what I've showed in the previous slides, the presence of contaminants, that's very common, and also the high human DNA in the blood that can affect the usefulness of your sequencing data and the sensitivity. The need for bioinformatic analysis, but there are a few companies out there that have their own pipeline now, which makes it easier for people like me, and dedicated, facilities and sequences and kits and fridge to support all of this work. And also, if you're starting this out and optimizing it, it is quite a bit of money. So we still have a long way ahead of us, and that includes trying out with more spiked and clinical samples. We are planning to do shotgun sequencing soon, and also trying out with the microbrain host Devin depletion filters, and also doing a bit more of the hybrid capture sequencing with our custom bacterial panel designed by Twist. And we also want to include other bacterial species like Burkholderia pseudomelia, which is very prominent in northern Queensland, and also these following ones that are very clinically relevant when we're talking about bloodstream infections. So ultimately, we just want to develop a methodology for rapid diagnostics directly from blood and hopefully have it clinically implemented to improve patient outcome. So just want to thank everybody involved. It's a lot of teamwork and coordination between the clinical side and the lab side. And yeah, thank you for listening.